So cutting with that two restriction enzymes will produce three fragments. Okay. And from the three fragments, we want these fragments to be ligated. And these two we don't want. Our plasmid also needs to be cut with the same blunt end cutter. Okay. So the plasmid DNA, when we cleave, they will also produce that blunt end. Now, these fragments can be either joined to this direction in the middle or in this direction. Okay. Therefore, this is not specific. Okay. This is non-specific. Okay. This slide shows some examples of the restriction enzymes and which type of sticky ends they produce. BAMase1 produce 5' staggered end. BGL2 also produces 5' staggered end. EcoR1 same. HIN3 also the same. HPA1 is a blunt end cutter. KPN1 produces 3' staggered end. Okay. SAL1 produces 5' sticky end. SAU3A produces 5' sticky end. Okay. So SAU3A on is compatible with the BAMase1. In the previous chapter, you heard about it when you learned the different cloning strategy. Different strategy using different vectors. Okay, now type 3 restriction enzymes. Type 3 is composed of two subunits. Okay. Similar to the type 2. Type 1 is composed of three subunits. Okay. One subunit is called M subunit, is responsible for recognition and modification of the DNA sequence. And the R subunit has the cleavage action. Okay. This enzyme, type 3, requires magnesium ion and ATP as their energy source. Okay. Some examples of type 3 are HPA1, MBO3, F4K1, etc. This enzyme has symmetrical recognition site and cleave DNA at a specific non-palindrome sequence. Okay. But it does not produce distinct bending pattern. Because to cleave the double-stranded DNA, two sites in opposite orientation must be present. Okay. Two sites. Which sites? The recognition site. Two recognition sites must be present in opposite orientation in double-stranded DNA. One strand of double-stranded DNA is cleaved about 25 to 27 BP away from the recognition site. That's why it does not produce distinct binding pattern. Okay. Therefore, this enzyme is not usually used in recombinant DNA technology. The type 4 has not been discussed further because type 4 is only used to work with the methylated DNA. And methylated DNA is not usually used in cloning and in genetic engineering. Therefore, it is not required for discussion. Okay. Now, application of restriction endonucleases. Different applications. They are applied in genetic engineering. They are used in the process of gene insertion into the plasmid vector, and that is very helpful in the cloning and expression of the proteins. They can also be used to distinguish gene alleles by specifically recognizing single base changes in DNA. That single base change is called SNP, single nucleotide polymorphism. Okay. But to distinguish this polymorphism, there must be a mutation at the restriction site in the target allele. Okay. If there is no mutation at the restriction site of target allele, then we cannot use this restriction enzyme for detection of the SNP. Okay. The restriction enzyme endonucleases or restriction enzymes simply remember when we say about the restriction enzymes, it solely represents restriction endonucleases. Okay. So restriction endonucleases can also be used for RFLP analysis, restriction fragment length polymorphism, okay, to identify the individuals or strains of a particular species. Okay. RFLP is a technique that is used to exploit variations in homologous DNA sequence. Okay. What does it mean? It means that we can use this technique to get the different fragments in an unknown genome or in a homologous DNA that come from the different locations of the restriction enzyme sites. Okay. And based on the fragments size, we can easily identify the position of the restriction sites. On that way, we can determine the location of the restriction enzymes. And in that case, we do not need to have the whole genome sequence. So this technique is very helpful in genome mapping. Okay. This tool is very helpful in genome mapping to locate the genes for genetic disorders, to determine the risk for different diseases, and for paternity testing. Okay. Even we do not have any information about that genome, we can easily map the genome. We can pinpoint the location of a particular gene in the genome, and we can determine the variations by analyzing the cutting pattern. And through that variation determination, we can predict the risks for diseases. And also, we can use that technique for DNA fingerprinting. Okay. Paternity test is a part of the DNA fingerprinting test. Okay. In RFLP analysis, the DNA is broken or the DNA is digested by the restriction enzymes so that it produces different pieces. Okay. Depending on the number of the restriction enzymes present in a given DNA or in a given genome or in a given sequence. Okay. Then gel electrophoresis is performed to analyze that different pieces or different fragments. Okay. On that way, we can get information of an unknown DNA or an unknown genome. Even we do not have any other information except the length. Okay. RFLP technique was first used in DNA profiling. Okay. But now this RFLP analysis technique has been less utilizable. Okay. Due to the rise of inexpensive DNA sequencing technologies. Okay. So RFLP analysis technique is now has taken place by the DNA sequencing techniques. And DNA sequencing is very cheap now. and very fast. Okay. RFLP analysis takes long time. Therefore, this technique has been obsolete. Okay. However, it is still used in some laboratory. Those do not have enough fund or enough resources. Okay. This slide shows an example of RFLP analysis to get the information about the genome. Okay. We did not have any information about the genome except the length. Okay. For example, this is genome. We only know through Jerry process that this genome has length of 4 kb. Okay, has a length of 4 kb. Now we need to identify the restriction size. So that we can have at least some information about the location and position of the restriction sites. Okay. Since we do not have any information, so we need to digest these unknown DNA fragments by defined restriction enzymes. Okay. So let's say this DNA is digested first with EcoR1. After digestion, we run the jelly electrophoresis and we find that there is only one band. Only one band. That means there is no EcoR on site. Then we digest with, let's say, Hind 2. Hind 2 or Hind 1. After gel electrophoresis, we get the same results. That means these sites are not present. Okay, so we need to digest with the different enzymes. In the next round of digestion, let's say Hind 3 has been used. Okay, so this is the genome, and Hind 3 is used to digest. Now, in the agar gel, after digestion with the Hind 3, we got 2 fragments. That means there is a Hind 3 restriction site. Okay. But where they are located, we can easily identify by the distance of the band. First band has 2.8 kb length. Second band has 1.2 kb length. So total is basically 4 kb. That means from number 1, okay, because 0 means nothing. It starts from number 1. From number 1 nucleotide to 2.8 kb nucleotide, the HIN3 enzyme restriction site is present. The next band is 1.2. So the rest part is 1.2. That means there is only one HIN3 site at this position, 2.8 kb. 2.8 kb position, the HIN3 restriction site is present. In the next round of digestion, let's say BAM-ACE1 has been used only. First time digestion has to be performed by the single restriction enzymes. Okay. When we digest with the BAM-ACE1, we get 3 bands. Okay. One is 1.8, another is 1.3, another is 0.9. So total 4 kb. That means there are 2 restriction sites in this DNA fragment or in this genome. We can easily locate their position. The first band length is 1.1. That means the first restriction site is located at this position, 1.1 kb position. The second band is 1.3 kb. Okay, 1.3 kb. So if we add these 2 bands length, that becomes 3.1 kb. So the second restriction site is located at 3.1 kb position. Okay. And the rest band is 0.9 kb. So that properly balances the total length. Okay. Now, we do not know whether this HIN3 is located between these 2 BAM-ACE1 or outside of this BAM-ACE1. So, to pinpoint the position of these restriction enzymes, we need to digest this genome or these DNA fragments by these 2 enzymes, HIN3 enzymes and BAM-ACE1 enzymes. Okay. And in the agar gel, we get 4 bands. So analyzing these 4 bands, we can easily pinpoint the location of all of this restriction site. Okay. With the double enzymes, the first band is 1.8 kb. So that corresponds with that 1.8 kb. That means the first restriction site is 1.0 kb distance, and that is BAM-ACE1. Okay. Now, the second band is 1 kb. Okay. Now, if we add this 1.8 and 1, it gives 2.8. So that corresponds to this one, 2.8. Okay. That means at the position of the 2.8 kb, the hint 3 is present. So that is the confirmation of the hint 3 position. The next band is 0.9 kb. Okay, 0.9 kb. Now, when we digested with the BAMASON, we got 0.9 kb. So, this is the 0.9 kb. That means it assures that the second BAM-H1 site is located after the hint 3, because the fourth band is 0.3 kb. Okay. And when we digested with the BAMASON, we got 3 bands. And when we digested with the hint 3, we got 2 bands. Okay. Second band length was 1.2. So 9.9 plus 0.3 corresponds to 1.2 kb. That means this distance is 1.2 kb. When we digested with the BAMASON, we got 0.9 kb here. Okay. And we got 2 bands, 1.8 and 1.3, that corresponded a total of 3.1. Okay. So this last band, 0.3 kb band, must be present between the hint 3 and BAMASON. So this length, this is 0.3. Okay. So through analysis of this banding pattern, we can pinpoint the position of the restriction enzyme site with their correct location. Okay. And this is called genome mapping. In this type of genome mapping, we do not know, we do not require any prior information of the DNA. Okay. Is it clear?
Yes, sir.
Okay.
Yes, sir.
Now, electrophoresis. Because agar gel electrophoresis is a required component in RFLP analysis. Okay. Electrophoresis is a technique to separate the molecules. That molecules could be protein, could be DNA. Okay. Based on physical characteristics such as the size, shape, and isoelectric point. Based on that physical characteristics, we can separate proteins and DNA through gel electrophoresis. Okay. Now, in the gel electrophoresis, we use two different compounds. One compound is agarose, another compound is polyacrylamide. Agarose is used to separate the DNA. Okay. Agarose is used to separate the DNA that has a range between 100 BP to 20,000 BP. Okay. Above 20,000 BP, we need to use different technique. It is called pulse field gel electrophoresis. Okay. In our lab, we have that pulse field gel electrophoresis. Okay. That is for the above 20 kb DNA fragments. Now, below 100 kb, below 100 BP, not kb, below 100 BP DNA fragments, agarose gel electrophoresis does not work. So in that case, we use polyacrylamide. Okay. Polyacrylamide gel electrophoresis can separate a smaller DNA fragments, even with 5 base pair length or 10 base pair length. Okay, even with the single base pair, we can detect using polyacrylamide gel electrophoresis. Basically, polyacrylamide gel electrophoresis is mainly used to separate the proteins. Okay. But this is also used to separate the DNA that has a small length. Okay, now in gel electrophoresis, the separation is achieved by moving the negatively charged nucleic acid molecules through the gel matrix with an electrical field. Okay. Nucleic acids are basically negatively charged. Okay, that could be the DNA, could be RNA. They have negatively charged. So in the electrical field, the DNA or RNA will move towards the positive charge. Okay. So that is the basic principle. Now, the rate of the migration of DNA molecule or protein molecule is inversely proportional to their molecular weight. Okay. Small molecules will move faster than the large molecules. Okay. Or the large molecules will move slower than the small molecules. So they have inverse relationship. However, besides the size, the conformation of the DNA molecule also affects the movement in the gel electrophoresis. Okay. Conformation of a DNA, let's say plasmid DNA, that has not been cut with any restriction enzymes. So the plasmid DNA are basically round, circular shape. Plasmid's shape is circular. Okay. So that circular conformation will move with different speed. Okay. Most of the plasmid DNA is supercoiled. That means they are compacted. Okay. They are round shape, but there are many pores within that rounding. So that is called supercoiled. Supercoiled plasmid will move fastest than the linearized DNA. Okay. While the open circular DNA will move at the slowest rate. Okay. Now the question is what is open circular DNA? This is circular DNA, but they do not have that supercoiled nature. Okay. So that means, let's say this is a plasmid, circular, but it has many pores, supercoiled. This will move faster than the linearized DNA. Okay. The open circular DNA will move at the slow rate. What is open circular? Let's say a circular DNA, but there is no internal fold. So this is called open circular DNA. Okay. So this will move at the slowest rate. So the conformation of the DNA affects the rate of the migration besides their size. Okay. So this is the apparatus. It is called gel electrophoresis gel tank. Okay. This gel tank has positive pole and negative pole. Okay. Electricity has positive end and negative end. So we plug that positive side to the positive pole, negative side of the electrical wire to the negative pole. Before running the DNA, we need to prepare the agarose gel. So this is the agarose gel. Okay, this is the agarose gel. In the agarose gel preparation, a comb is used. Okay, and the percentage of the agarose varies. It varies from 0.5% to 1.4% agarose. Okay. And that percentage depends on the length of the DNA. Okay. So we place the comb. Okay. We place the comb. Then we prepare the solution by melting. Okay. When we prepare this agarose solution, we add ethidium bromide. So when they are in liquid form, after heating or after melting, we pour the solution into that casting chamber. Okay. So when they cool down, they become solid. Then we pull up that comb, and that comb creates the hole because there was no gel. And this hole that has been created by putting the comb is the place where we put our sample. Okay. To track the movement of the DNA sample is mixed with tracking dye. Okay. We add some dye in the DNA. Okay. That dye could be Komasi blue or any other dye. So that we can visualize the movement of the dye. Okay. The movement of the dye ensures the movement of the DNA. Now, after finishing the run, we need to visualize the band under UV light because we do not see the DNA band on the agar gel. Okay. That's why we add the ethidium bromide. Okay. Ethidium bromide binds with the DNA molecule and it produces fluorescence under UV light. Okay. So this is the band when we observe under UV light. Now, ethidium bromide can be added at the time of the gel preparation, or ethidium bromide can be added in the buffer. Okay. Either way, it is more preferable to add the ethidium bromide with the gel. Okay. So that it creates less messy and it is safer than mix them in liquid solution. Okay. So this was the sample loading hole that was created by placing the comb, and that DNA migrated from negative to positive because DNA has negative charge. So they will migrate towards the positive charge. Okay. When we apply the current. Okay. Now, to maintain the conductivity, after placing the gel, we add the buffer solution. Then we add our sample very carefully in that well. Then we run. That means we switch on to have the electrical flow. Okay. So these bands are a smaller band that have moved very fast. These bands are large band that have moved slowly. বুঝা গেছে?
জি স্যার।
Okay. Now, you have learned before that compatible enzymes, right? BAMH1 is compatible with SAU3A1.
জি স্যার।
So that compatible enzymes have another name. It is called isoschizomers. Okay, isoschizomers. They are restriction enzymes isolated from different organisms but have identical recognition site and identical cutting pattern. In some cases, isoschizomers recognize the same restriction site, but cutting pattern is different. Okay. So it will depend on the type of the restriction enzymes. For example, ASP718, KPN1, they have the identical recognition site and they have identical cleaving bases. Okay. For example, these two enzymes, they have been separated from two different enzyme organisms. Okay. You have learned the nomenclature, right? ASP means Aromonas species and KPN means Klebsiella pneumoniae. Two different microorganisms. Okay. But their enzymes are similar, identical. Okay. They recognize the same palindrome sequence and they cleave the phosphodiester bond between two guanidine. So this type of enzymes are called isoschizomers. Okay. As I mentioned, that some pairs of isoschizomers cut their target at different place, but they recognize the same palindrome sequence. For example, SMA1, XMA1. This is a pair. These two enzymes will recognize the same restriction enzyme, same palindrome sequence, but the cutting pattern would be different. Okay. Instead of, let's say, instead of cutting between C and A, probably they will be cutting between phosphodiester bond between two cytosine. So that will be the difference. Okay. Now, when we work with the digestion, there are some artifacts created. When the reaction condition is not optimal. Okay. The reaction mixture should have optimum concentration of the ion, optimum concentration of the DNA, optimum concentration of the enzyme, and the optimum concentration of the time to be used for digestion, together with the optimal temperature. Okay. So if the conditions are not optimal, then some artifacts are produced. That artifacts are called star activity. Okay, are called star activity. So what does it mean? Let's say this is a DNA fragment. Okay. And only one restriction site is present, echo R1. Echo R1. Echo R1 site is present. Now, after digestion with this enzyme, we will have only two bands. on the agarose gel. So we run the gel. This is the marker. And depending on the size, we'll be getting only 2 bands. Okay. We'll be finding only 2 bands. Let's say one band is here and another band is here, 2 bands. So that is under optimal condition. But if the environment, that means you have done something wrong, or you have exposed to cliff for a long time, or you have mistaken to provide the optimal temperature, or you have messed up with the calculation of the concentration of the DNA or enzyme or the ions present in the buffer. So at non-optimal conditions, the EcoR1 will show relaxations. That means instead of having 2 bands, it will produce many bands. Many bands. So if the many bands are appeared in this condition, that means the condition was not optimal. And this artifact is called the ester activity. বুঝা গেছে?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
What is the time now? Okay, we have time. Okay, now ligases, enzyme. Okay. Before going to the ligase enzyme, let me show you why do we need ligase enzyme. So let's say this is a plasmid DNA that has been digested with restriction enzymes. And this is the sticky end. Our target DNA has also been digested with the same restriction enzymes. Therefore, this end will be complementary to this end, and this end will be complementary to this end. And through that complementary base pairing, ultimately, it will have this type of circular form. Okay. This type of circular form. But there is still gap because no phosphodiester bond has been created. Phosphodiester bond is not spontaneously created. So this is called gap or nick. Okay. This is the gap or nick. That's why to create the phosphodiester bond, DNA ligase is used. Okay. DNA ligase is used to seal the gap, to seal that nick, so that they get the phosphodiester bond. If without phosphodiester bond, this plasmid is inserted into the bacteria, that plasmid will be degraded by the nucleases because they are more vulnerable because they do not have phosphodiester bond. Through creating phosphodiester bond, now they will not be easily attacked by the nucleases for cleavage. Okay. So that's why DNA ligase is required. Okay. Okay. So the main function of the DNA ligase is to form the phosphodiester bond between two nucleotides. Okay. And the joining process is called the ligation. Okay. And this is very much required. Otherwise, the damaged DNA, that means the DNA with the gap, will be degraded. Okay. Now, how this enzyme works? How does this enzyme works? How does this enzyme work? DNA ligase enzyme requires a free hydroxyl group at the 3' end in one DNA strand and a phosphate group at the 5' end of another DNA strand. Okay, so this is the condition. 3' end must have free hydroxyl group and 5' end must have free phosphate group. Otherwise, no phosphodiester bond will be created. And this process requires energy. Okay. This process requires energy. Now, there are different types of ligases. E. coli and other bacterial DNA ligase utilizes NAD as energy. Okay. Whereas T4 DNA ligase uses ATP. Okay. Now, ATP is more stable than NAD because this is the oxidized form of nicotinamide adenine dinucleotide. So this oxidized form is easily reduced in the solution. Therefore, it is very hard to work with this oxidized DNA. Therefore, T4 DNA ligase is preferred, is mainly used in the cloning experiments. বুঝা গেছে?
Yes, sir.
Okay. So this already I have covered and you have learned why DNA ligase is required. Okay. The next enzyme is polynucleotide kinase. I stop here today. I'll continue in the next class from this slide. কারো যদি কোনো প্রশ্ন থাকে করতে পারো. If you have any question, feel free. যদি কোনো প্রশ্ন না থাকে তাহলে সবাই লগ আপ করো. See in the next class.
স্যার একটা কথা ছিল.
Right.
স্যার একটা কোয়েশ্চেন ছিল.
Go ahead.
স্যার আপনি বলেছেন যে অনলাইন ক্লাসের সময় কোনো এক্সাম হবে না, তাহলে আমাদের সানডে পর্যন্ত যদি অনলাইন কন্টিনিউ হয় তাহলে কি আমাদের কুইজ নেক্সট অফলাইন যেদিন প্রথম ক্লাস হবে ওই দিনই হয়ে যাবে?
হ্যাঁ, আমি জানিয়ে দিব.
ওকে স্যার, থ্যাংক ইউ.
I'll place a notice in the Google Classroom. Okay, সবাই লগ আপ করো. See you in the next class.
Sir,